Okay, guys, we're off to the second half of this lecture video or this lecture series, and instead of GMP, good manufacturing practices, we're going to be focused on GLP, yeah, you know me, good laboratory practices. So the first half of this was all about the manufacturing side of a company, and that company could be a drug company, it could be a food company, it could be a cosmetic company, it doesn't matter, but someone that, in our point of reference, that the FDA kind of regulated, that they controlled somewhat, pushed out some rules and regulations on what they should be doing at that particular location. And it's all about the manufacture of that product. Now we're actually going to turn and we're going to look at the laboratory side of the house and not the manufacturing side of the house, and this is where GLP comes into play. Now the GLP, I want you to understand that there could be a very tiny c that happens in front, and that tiny c means current, just like CGMP meant we can have CGLP, because these rules or these regulations can actually change and get updated over the course of time, and we should be making sure that we're following the CGLP regulations that are enforced by a third party. And that third party could be the FDA, again, if we're drug, cosmetics, food, or it could be another regulatory agency like the EPA if we were working on the environmental side of the house. So again, some of this stuff you're going to kind of seen before. It's a repeat, it's review. But there's going to be little nuggets of information that are somewhat new over the course of time as well. So the first thing that we need to talk about with GLP is like why, right? Why on earth do we got to follow these awful rules and regulations and make my life so miserable? Why can't we just do some testing in a lab and just send it off and be okay with it? I mean, they trust me, right? Do you trust me? Maybe that's the bigger question. So GLPs are going to be implemented for a number of reasons. The federal government wants us to have a minimal standard in a laboratory setting, and that minimal standard should help us to determine two things. And one of those things is our safety, ding, 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 and the other one is the efficacy, ding, 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 ding. Those are the things that we want to pay attention to when we handle these products throughout the time. So GLPs are going to help the lab and the workers in the lab to demonstrate the safety of what has been manufactured or what is getting used in the manufacturing process, and efficacy. So that means if this is a finished product, then that finished product better do a job. And efficacy is all about that job. Does it do its job? Yes or no? That's what this is all about. So those are the two major players as far as why we do GLPs to begin with. Now, I want you to keep in mind that at a certain point when these GLPs were recommended and when regulations were built around them, this is going to help these companies, whatever company, again, whatever industry that we're working in, it's going to help them to give birth to the QA and the QC departments, the quality assurance and the quality control, because that's what the purpose of QA and QC is all about, right? And that's why we're taking this class. It's all about the assurance of those ingredients or that product and the control. What happens to those things while they're under our roof and either getting manufactured or getting tested by a laboratory worker. So QA and QC is going to come out of this, and QA and QC is going to help ensure that good laboratory practices are in fact recorded, and that people are doing them, and that we've got a tracking system in place, so that way if an auditor later on down the road comes into a laboratory and they want to take a look at our work, then we have the proof in the pudding. And we can give that to them and say, have fun, see you later, I'm going to go do my job. Now you do yours for days on end, and let me know if you have any questions. Bye-bye. And I can feel really good about what I've given those auditors because I've done my job the proper way according to the CGLP regulations in place. So, yet again, I want you to understand that, yes, this is cosmetic, drug, and food product related. I always reference, it seems like, the drug industry because I think that maybe people take more interest in that in the beginning than some of the cosmetics and the drug or the cosmetics and the food. But it applies to everything. And we'll talk a little bit more about that when we start giving, I'm talking about myself in plural, when I give you a little bit of timeline that begins to happen. Notice here, though, cosmetic, drugs, and food, that's really more in the area of Food and Drug Administration, the FDA, but we do have other third-party regulators like the EPA and sometimes the FDA, depending on what the material is, but we also have environmental types of products that are going to be tested, that are going to be performed in a laboratory with GLPs in mind as well. It's not just the FDA, but really fair game, to be honest with you. All laboratories should be operating in this particular way. So like GMPs, GLPs are going to focus on a few things, and here I've listed a few of those for you already. So those are going to be training records, laboratory space, equipment, reagents, and chain of custody. This is nothing new, right? It's not surprising either. If the manufacturing side of the house has to control this and has regulations in place where they have to document, they have to provide information to auditors and those types of things, the laboratory, the proper laboratory, will probably have to do this as well. So that's not surprising. No one is out of the weeds of the regulations. So we're all in this together. It's all the same company, the same mission. So of course, we're focused on the same general areas. So training records include everyone that has been hired for the laboratory side of the house. And what was their training? What was their education? When did they complete their training modules? Who signed off on it? Do you keep a record of those? So very often we start off with little bits at a time, and then we keep going up and up and up and up and up, and the file gets bigger and bigger and bigger and bigger and bigger. Next up is the laboratory space itself. So I'm talking about the physical laboratory space. We saw that on the GMP side of the house. We're going to see it on the GLP side of the house as well. So an auditor will come in and they'll start looking around at these laboratory spaces. They'll start investigating these laboratory spaces, everything from the ceiling down to the floor. There are certain criteria, certain things that they're after, and they want to make sure that those things are in place when they come around and do their job. Next up is the equipment. If they do that on the GMP side, they're also going to do it on the GLP side. So the laboratory equipment. So these are things like our HPLCs and our GCs and our density meters and our polarimeters and all of those types of things that we would be using on a daily basis. The analytical equipment. The manufacturing, those were much bigger pieces most of the time. Those were big vats, big mixers, big blenders, big processors. The equipment here, most of the time, is a little bit smaller, fits on a lab bench, but those things are going to be audited and inspected as well. Just like the GMP side of the house, the GLP side of the house will need to keep track of reagents. So reagents of what's getting used in the laboratory side to do that testing with. Things like expiration dates, when did they get ordered, who came in contact with them, including chain of custody forms. So what that means is that a chain of custody is from the moment of intake to the moment of disposal. That's what chain of custody is all about. Intake meaning when it arrives at the company. So let's say UPS brings me the box and I've got a reagent bottle on the inside. Well, whoever opens that box begins the chain of custody because they have been first contact with that bottle by the time that it arrived at the facility or at the company. So the intake process is going to typically initiate the chain of custody, and then all of the testers that begin to use that material, maybe over the course of months, maybe over the course of years, who knows, but anyone that's using that material from that original bottle will have a chain of custody form that they need to fill out, almost like a library checkout card back in the day when we had those, right? Who has it? When did they get it? When did they use it? When did they bring it back? That's what this is all about. And then that will go all the way into disposal. So by the time that we throw that away, who did it? How did they do it? And why? Was it just past the expiration date? Or was it contaminated? What was the reason that we actually disposed of this material to begin with? Maybe it was an empty bottle, but still a chain of custody form is going to follow that material through from intake to disposal. So with GLP, or good laboratory practices, I want to talk about a few timeline scenarios that begin to happen here that I think it's important to at least mention to give you a broader idea of why certain policies and certain regulations were put in place. So back in 1906, we mentioned this a little bit, somewhat the creation of FDA in a way, and this is from the Pure Food and Drug Act. Notice cosmetic was not part of this at all. That begins to change a little bit, 2 or 3 decades later, but the Pure Food and Drug Act really only focused on food material and drug material, of course. So the pharmaceutical companies and the food companies. Now, the Pure Food and Drug Act mainly actually at that particular time focused on food, because that was really what they were seeing most of the time. And the regulations and the restrictions and those policies about drugs really weren't enforced like they are today, or at least there weren't as many of them. So 1 year later, 1907, we get this material called sulfanilamide. And sulfanilamide was really the first antibiotic that was produced. This was typically in a powder form, and sulfanilamide works wonders for things like strep. So your first antibiotic that can be given to a person. And this was used for a number of years. 1907 all the way up until 1930, when we find a scandal. So 1930s, meaning about the mid-1930s, 35, 36, 37, somewhere in there, we begin to see a problem with sulfanilamide. And we'll talk about this in just a second, but this was a big red flag, and the United States had to figure out how on earth can we prevent this scandal from happening again. So 1930s, we get a red flag, we get a problem, and that initiates a 1938 change. And in 1938, we get the FD&C Act. And the FD is food and drug, and the C is now cosmetics. So now cosmetics falls into the discussion. But the FD&C Act, well, the United States says the manufacturer has to prove the effectiveness of a drug. Oh my God, are you kidding me? They have to prove. You mean that up until 1938, companies could be producing products and they didn't really have to prove or submit any type of information about the effectiveness of the product that they were making? You mean, how effective is it? Like, if I take this, will it do what it says that it's going to do? Absolutely. That's exactly what I'm saying. So 1938 was the first stab in this process. That company has to prove the effectiveness. It is not the job of the government. It is not the job of the FDA. It is not the job of a third-party organization to prove your effectiveness. It is your job as a company to prove the effectiveness of that product when you submit that material for approval at the FDA level. So that sounds great, right? Well, a couple of years later, look at this, 1941, we get sulfathiazole. Oh, here we talk about another red flag that begins to happen. We'll discuss this in just a second, but it seems to be another problem. And then about 20 years later after that, finally in 1962, we get another amendment that begins to take place. And it says, oh, hold up now, hold up. 1962, we're going to push in a new revision. And that revision is saying, yes, we're going to check the effectiveness like you have been up to this point. But there's something else that you got to do, folks. At a company, at a facility that's making things and making products to sell to a consumer, we want to make sure that you're doing some tests on safety. Oh my God, are you kidding me? You mean up until this point, they did not have to provide safety studies? They didn't have to kind of do any type of study or report or submit data about how safe the product is if a consumer tested? Well, kinda, but not to the extreme that they began to want in 1962. So in 1962, the term safe and effective began to get used. Then 8 years later, 9 years later, in 1970s, we get some more problems that begin to happen here. Notice it says numerous cases of malpractice surfaced in toxicity testings. Well, wait a minute. Just a handful of years ago, I thought that you were supposed to do safety tests. Well, they were. At least they thought they were. And now all of a sudden you get problems with toxicity testing. Wouldn't that actually fall into the same category? It should. But you're putting too much faith in these companies. You're putting too much faith in the laboratories to do things the right way like they're supposed to. So we're going to talk about a notorious example here of a laboratory that really was reporting data, but were they reporting proper data? That's something that we're going to have to talk about a little bit later. Next up, 1976, we see FDA finally proposes what they call GLP. So good laboratory practices began to get pushed out. This is the way they want laboratories to run. This is the basics or the minimums of what have to be checked off. And then you can go above and beyond that at your site location if you want. But these are things that we're going to be scrutinizing when you begin to work and make and manufacture and test these types of products that the FDA will begin to oversee. Next up, 2 years later, the GLP regulations are finally published. So at this point, they finally go out because 2 years prior, they were talking about them, they were assembling them, they were writing them, they were kind of putting them in law. They were letting these laboratories know that these were going to be up and coming to get ready for them. And finally, in 1978, they become in place on the books and they get pushed out to all of the laboratories that are related to the FDA. A handful of years later, in 1983, the EPA finally gets on board. Oh, well, finally we get another call letter. Environmental Protection Agency, right? So the Environmental Protection Agency finally looks at these GLPs and they go, you know, these are pretty good. I think that we can utilize these GLPs in our area as well on the environmental side. And that's exactly what begins to happen in 1983. So the EPA pushes out their GLPs, which are very similar, if not identical in many ways. They could have a few different things that they look for, or maybe not as restrictive, but overall the name of the game is the same. Good laboratory practices. So that's where this video is going to stop. I'm now giving you a timeline, what kind of leads up to the early 1980s, which was a great time for music and TV and everything, folks. You just don't know what you missed out on. But for those of you that were kind of up and cranking in 1983, right? All right, so from this moment, we'll, in the next video, talk about more details of this timeline. What are some of the things, the big kind of red flags that begin to happen in the timeline, and some details about, you know, why were these things so bad? I mean, you know, they had to be something major for the government to step in and be like, hey, we need to do something to fix these problems so it doesn't happen again. And that's one of those things that will happen and that we'll talk about in the next video. So don't take a break. Just hit play on the next video and keep trucking. You're doing so good. I mean, you're listening to me. You're writing down notes. You're listening at me at 2 times the speed, and I sound like a little stupid.